Thanks a lot for the introduction. My name is Susanne Bornelöv, and I'm a group leader at University of Cambridge, and I will present this work, which is some of our most recent work. And this was done together with and led by a postdoc in my lab, Titrai Dash. So just to put everyone on the same page. So we have a lot of genes, and at a given time, some of these genes are expressed at a high level, and some of them are expressed at a low level, and some of them might not be expressed at all. And what determines that is really, okay, need to learn this, short sequence motifs in the promoter region that is located just upstream of the transcription start site. So we have transcription factors, which are specialized proteins that bind to and recognize specific motifs or like near matches of motifs. So we have, for instance, the green transcription factor here binding to the green motif, and that is leading to an activation of gene expression. And similarly, the binding can lead to repression of gene expression, but most of the time we probably have complex interactions or competition between these transcription factors. So it could be like the green and the blue bind together, they inhibit the yellow one, and this leads to a strong activation. So how do we study this? So most experimental approaches are insufficient. So like you might knock down your transcription factor of interest and measure what happens, but it doesn't tell you anything about this competition or cooperation. So, and I probably don't need to convince you. So what we really need is data-driven approaches. And to do that, what we ideally would like to have is a large data set with a lot of promoter sequences that have these kind of different motifs in different combinations paired with expression values. So if we just think about how much data we need, so humans have 1,600 different transcription factors. And if we want to have every combination of two transcription factors and also like in every ordering, that would be 2.6 potential interactions. But the human genome only have 23,000 genes. So even if we take every promoter as part of our training data, it's easy to see that it wouldn't be enough. So what we really need is to go beyond naturally occurring variation. So what has really driven this forward is massive parallel reporter assays, as you heard about already. So this is an example from budding yeast. So in this case, they used 80 nucleotide random sequences, which were inserted into a minimal promoter construct that was located upstream of a yellow fluorescent protein. And this reporter also contained a constitutively active red fluorescent protein, and that allowed them to grow this yeast and to sort the cells by the fluorescence color into 80 different bins. So, and then these bins were sequenced separately, and then you can quantify the number of times a particular promoter, a particular random sequence, is occurring in the different bins. So if you have a promoter that is occurring half of the time in bin 4 and half of the time in bin 3, we would say that it had expression 3.5. And we really got involved in this through the Random Promoter Dream Challenge that was read by Rafi, who just had a brilliant talk. And the task here was to predict gene expression using millions of random promoter sequences. So the participants were given one of these data sets that I just described that had 6.7 million promoter sequences and the associated expression values. And then every team went and they built their own model. And in the end, all these models were compared against each other across 8 different categories of promoter sequences. So for instance, high expressed sequences, low expressed sequences, or native yeast sequences. And we took part in this challenge, and we did quite all right. I'm not going to go into the model in too much detail, but this is the model that we submitted in the end that had 6 convolutional blocks and residual connections going over every other block. And then the output of the last convolutional layer was flattened linear blocks and eventually produced a single prediction as the expression value. And this is relatively easy to predict. So this is the output of our model. So we have on the x-axis, we have the true expression, and on the y-axis, we have the predicted expression. And as you can see, it follows kind of a nice diagonal line. And this is across the data that we didn't have access to while developing the model. And as I said, the models were evaluated across different categories of promoters. And we did well across all of these categories. That took us to 4th place in total. There was a benchmark model also in the competition that would have gotten 20th place as a comparison. So there was a quite significant improvement, both from us and from others against the previous state of the art. So what's quite interesting here is also to look at the performance in terms of Pearson R and Spearman correlation across the different promoter sequences. So for instance, we performed best across random promoter sequences, which makes sense because that's the type of data that was used to train the model. And we performed worst across high and low express sequences. And these were sequences that had been in silico evolved to have unusually very high or very low expression. So they went well beyond what you can see in native yeast sequences and also beyond what was available in the training data. So it kind of makes sense. So just looking a little bit more in detail how we did against the previous state of the art. So the highest improvement was seen across a category called challenging sequences, where, so the baseline here is the Vaishnav-de Boer model, and we had a 57% relative improvement in performance against that previous state of the art. And the winning model had a 76% improvement. So challenging sequences in this case was defined as sequences where a previous biochemical model had the biggest disagreement with a previous transformer model. So kind of sequences where previous models disagreed with one another. But I guess the models in this challenge didn't particularly have the same disagreement or weaknesses. So the category where we saw the least improvement was single nucleotide variants, where we had only an 8.2% improvement over the previous state of the art. And this was defined as pairs of sequences that differed by only a single nucleotide. So the task was to predict the change in expression or the difference in expression between these two sequences. And that, of course, was not particularly part of the training data. So it seems like if you train your models on random sequences that are very different from one another, it might still be hard to predict exactly the effect of single nucleotide variants. So to make the comparison a little bit more fair, so this baseline model was originally trained for a different data set, a larger data set. So to make it fair, we also retrained our model for that larger data set to make the sort of reciprocal comparison. So the performance of the previous model here is slightly lower than the performance of our models. That shows that our model architecture is stable. It can be applied on other data sets, and it is state of the art or close to state of the art. So just to wrap up so far, so I have shown that yeast gene expression can be predicted from promoter sequences, and I have shown that we have a model that is state of the art or near state of the art for this task. And I would now like to spend the last 7 minutes sort of trying to convince you why we should care about this and what we can do with this type of models. So going back to these data sets, so we took all the sequences from the training set, 6.7 million sequences, and we sorted them from high to low expression. And if we then take the 10% highest expressed sequences and the 10% lowest expressed sequences, and we did a motif enrichment analysis in these. So this can roughly be interpreted as at the top being motifs contributing to activation of gene expression, and at the bottom being motifs contributing to repression of gene expression. But this still doesn't tell us anything about this cis-regulatory logic. So to address that, we looked at sequences in which the motifs co-occur. So I will walk you through this in detail. So the heat map is showing the average expression in the training set for sequences that have the indicated combinations of motifs. So first of all, across the diagonal, we have the average expression of sequences containing only a single motif. And whenever you go away from the diagonal, it's the average expression if you take the two different motifs from the x and the y-axis. So in the upper left, for instance, we have REB1 and ABF1, which both on their own contribute to high gene expression. And together, you get an even stronger increase in gene expression. So it shows that combining activators leads to higher activation. And if you look at the bottom right, we have the same thing, but the opposite. So if you combine repressors, you get an even stronger repressive effect. But this is still, it kind of looks like additive effects. So we were interested in whatever deviates from these additive rules. So to do that, we subtracted kind of the diagonal. So whatever you have from a single transcription factor per row, and I will walk you through what this means. So this is the figure to the right. So the first column is shows the change in the predicted expression if you add Reb1 to a sequence that already contain any one of the indicated different motifs. So essentially, adding Reb1 to Reb1 doesn't make a difference. That's the top left. But if you add Reb1 to anything else, you see a strong increase in the predicted gene expression. So Reb1 is a consistent activator regardless of the previous sequence context in this case. And to the right, we have YAP7, which has a similar repressive effect. So if you add that, doesn't matter what your first motif is, but if you add that one, you get a consistent repression. So the interesting here are the kind of exceptions to this rule. So we have UME6, which on its own, when you add it to most motifs, doesn't make any difference. But if you add it to a sequence that already contain a PDR3 or an IME1 motif, then it suddenly has a strong negative effect on predicted gene expression. So we wanted to look into this a bit further. And the first thing we did, it's getting dark, was to look into how does the actual motifs look like. So this is the motifs from the Jasper database. And as you can see, the IME1 motif is overlapping with the UME6 motif by 5 nucleotides. And then we switched from the training data to the test data. So the figure to the right is showing the overlap, or how many random sequences in the test data that contain one motif, the other, or both of them. So there are 368 sequences containing both of them, and presumably many of these are probably overlapping. So we then went back to our model to try and understand how does this sort of competition interaction work. And we went and used in silico mutagenesis, which I think you heard about a few times. So the idea here is you take your sequence and you mutate every possible position in your sequence, and you introduce every possible mutation, and you measure what is the change in the predicted gene expression. So for instance, the negative C here means that if you mutate this C to an A, G, or a T, we would get on average 4 units higher gene expression. And 4 units here would be like taking something from bin 10 to bin 14 in this 18 bins experiment. So like it's a huge shift. And this ISM analysis, it sort of highlights different motifs. And the interesting thing about this particular promoter is that to the left, we have the IME1 motif on its own, which is not predicted to have any particular effect on gene expression. And in the middle, we have the IME1 motif and the UME6 motif overlapping, which is predicted to have a very strong negative effect. But this is just one sequence. So going back to the entire test set and looking at it across all the sequences. So this heat map shows you the ISM scores across all the sequences that contain at the top the IME1 and the UME6 motifs, and I'm now requiring them to be overlapping. And the heat map is centered around the IME1 motif. So what this is showing is that our model is interpreting this overlap between the two motifs as being a strong negative predictor of gene expression. And at the bottom, we have sequences containing only the IME1 motif, which is predicted according to our model to have either a neutral or a weakly positive influence on gene expression. So we have this kind of switch-like behavior where IME1 on its own is neutral or weakly positive, and if you add a UME6 motif partially overlapping, it becomes strongly negative. So it might be either sort of competition between the two transcription factors trying to bind in the same place, or it might be some sort of interaction between the two proteins. So just to wrap up, so we have shown that complex cis-regulatory logic can be studied using deep learning, and that we can use this model to provide hypotheses about interaction effects. And we have identified this potential interaction between IME1 and UME6, which might be either competition or like interaction between the proteins. So with that, I would like to also thank my lab, and in particular, Titarai Dash, who was the person, my postdoc, leading the project, also Mariana Trapozzi and Fredrik Svensson for contributing to developing the original model in the DREAM Challenge. And this study was just accepted in Bioinformatics Advances, so you can use the QR code if you want to read more about it. And thank you all for listening, and I'm happy to take any questions.
Thank you very much, Susanne. Questions?
Thank you. I wonder, have you or anybody looked at the orientation of motifs with regard to effect on gene expression, so forward, reverse complement?
In this case, in this figure, for instance, I have reverse complemented it if the motif was on the negative strand. So the -1 to -10 is always upstream in sort of the direction of, well, how the motif is annotated in the database, I guess, but it always is the same direction. So yeah, I guess that in terms of the ISM score, yes, there is a directionality because you have like this position, for instance, that has an effect. And if it was mixed up, it would be a little bit of each.
Thank you.
I would have another question, because I also work a lot on prediction of expression. And one of the things that always is a problem, I feel like, this steady state expression, right, which also includes post-transcription regulation. Now, that may not be so common in yeast, but I wanted to hear your take on this, in the way it's set up and the way you did it. Oh, this was MPRA data, right? This is not expression. But if we're predicting from gene expression and then we're doing these kinds of analysis, then of course we may underestimate the post-transcription effect. So what's your take on this? Is this concerning? Do we, need to do this differently? Or?
No, it's quite exciting, I think. I mean, ideally, we would want to measure both like the protein level, but also like the mRNA stability and the actual translation rate. In this case, only measuring the YFP levels, only measuring the protein level. So of course, it could be that a certain variant is somehow triggering like immune-like response and degrading the RNA, but we would be blind to it in this case. But I would love to measure it. I guess to the previous question also, I mean, if you mean the directionality of the motif relative to the transcription start site of the gene, I think in this case, I mean, as I said, I had to flip them to sort of make the heat map making sense. And I think that means that the motif has the same effect in this case, in both cases. But I guess for other motifs, there might be a directionality component to it, but not to IME1 and UME6, I think.
All right. When there's no further question, thank you very much, Susanne.